Okay, everybody, welcome to another WBESS episode. Just as a quick recap, WBESS stands for Western Biology Evolution Seminar Series. I'm your host, Mateus, a PhD student in biology here at Western. And today I have the pleasure to have Dr. Laura Tensen with me. Dr. Laura actually she just started her group in Germany, so I will let her tell us a little bit more about that. But she will be presenting to us her work with South African leopards and the population genetics of those groups. So I'm very excited to have you here today, Laura. Thank you very much for being here with us. And with that, I will let you take the floor.
Yes, thank you, Mateus, for the invite and for the introduction, and welcome to anyone who is going to stream into this talk. Yes, I'll be talking a little bit about leopards today, selection drift and the evolution of phenotypic variability, particularly with a focus on leopards that occur in the southern tip of South Africa, the Cape, we call it, where leopards are about half the body mass of leopards elsewhere. And we'll be talking about this striking red leopard that you see on that picture. So we know that phenotypic variation, whether it's color polymorphism or differences in body size, we know that they occur throughout the animal kingdom, and typically you'll see variation occurring throughout a species range. These visible traits, such as color polymorphism or differences in body size, they are of particular interest to evolutionary biologists because of their traceability in the wild, because they're visible to the naked eye. And as a result thereof, they can be used to test certain evolutionary theories, such as balancing selection or sympatric speciation. So sympatric speciation, perhaps you recall, is when species start to diverge while they still live in the same area. So there's no physical barrier between them that separates them. So we know that colomorphs can be subjected to strong sexual selection, which can lead to assortative mating. So assortative mating means that the chances that you mate with a species of your own kind, or colomorph of your own kind in this case, is higher than you would predict by random chance. So this can either mean that animals have an actual sexual preference for animals that look the same way, or it's more of an opportunity thing, which means that the chances of them running into a similar colomorph are higher. So they may also face disruptive selection. So this is when two colomorphs have different selective benefits, and this can result in spatial or temporal niche segregation. So we know, for instance, that black leopards, they tend to hunt more at night, and they stick to tropical rainforests more so than the wild type leopards. So this would be spatial and temporal niche segregation. As a result, it has been proposed that color polymorphic species, or another form of polymorphism, that these animals tend to speciate more rapidly due to niche segregation and assortative mating. So this has been proven in birds already, and we did a similar study to show that this is happening in carnivores as well. So quite a striking example of this niche segregation is the Asiatic golden cat. So there's 6 known color morphs that occur at slightly different microhabitats. Typically, you'll see that dark individuals move higher up the slopes. So this is where assortative mating could be shown quite nicely. So in this case, the chance of you running into a species or a member of your own species that looks the same are simply a little bit higher. So to put that into an overview again, just to show you how speciation rates could be linked to color polymorphism. So if you have color polymorphic species and you'll start to see niche segregation over time, this can lead to a speciation event if these species become reproductively isolated from each other. And eventually this can also relate to extinction events, in the sense that the theory predicts that if speciation rates go up due to niche segregation, then if habitat changes due to climate change, you could predict that across the whole phylogenetic tree, the chance that all species go extinct is also smaller. So the phylogenetic tree would look slightly different, where polymorphic species are ancestral to monomorphic species. Of course, monomorphic species also speciate. In this case, they might create spots over time as a habitat matching mechanism, but if the habitat goes, so does the species. So in a phylogenetic tree, it will look slightly different where one species evolves into the next. So this is one part of evolution. We know that selection is a driving force, but there's two other important driving forces, and that is gene flow and, of course, genetic drift. So genetic drift and heterozygote deficiency are also known to influence phenotypic variability in populations. So it has been suggested that recent changes in coat color and pattern might indeed result from population bottleneck, simply because effective population sizes decrease, increasing the chances that related individuals mate, which can increase homozygosity and thus the chance that recessive mutations become expressed. Particularly for carnivores, this might appear to be true because carnivores occur in much lower effective populations and they're very sensitive to habitat fragmentation. So what you'll often see in small effective populations that have become isolated from other populations, you might occasionally start to see these rare color variants popping up or other phenotypic malformations like kinked tail or asymmetric skull. There's been a couple of these different morphs observed in small populations. So to put that in an overview again, if you look at the left, this is what you commonly see across species that have a large range, is that you start to see gradients, in this case color gradients from north to south as a subtle habitat matching. In the middle is that color polymorphism. So this is the example that we gave of the Asiatic golden cat. So this is where different morphs are established throughout a species range. And these morphs are typically sustained through balancing selection. And on the very right, this is what we call a rare color variant. This is typically in these small effective populations where occasionally a rare color variant pops out. And there's different genotypes associated with these three groups. And I think the main message is here that typically what you would expect with rare color variants is that it's rare recessive alleles being expressed when they start to occur in homozygosity, typically due to the effect of inbreeding. So this is just an overview of rare color variants that have been spotted in the carnivora family. Quite a few different ones, cinnamon, leucistic, diluted forms. What maybe stands out is there's quite a few sightings in India, and the Atlantic rainforest area is quite known to be associated with habitat fragmentation. But I just wanted to show that some of these examples are quite striking. This is the black Canada lynx. We have a leucistic puma, a spotless cheetah, and we have the pseudomelanistic tiger. So this pseudomelanistic tiger occurs in one reserve in India and currently reaches frequencies of up to 78%. So we know that small effective population size does have phenotypic consequences on extant wildlife populations. The high frequency, so we try to measure the average frequency of all these color variants that occur throughout the carnivora family tree across the globe, which was on average 20%, which is pretty high, and it seems to imply that genetic drift is locally overruling natural selection. So in this sense, some morphometric and phenotypic changes can be added to the list of the consequences of inbreeding on wildlife populations. So now I want to take you to quite a unique case study at hand. So this is leopards from the Western and Eastern Cape of South Africa. So this leopard was always known to be smaller than leopards up north, so about half the body weight of leopards in other areas of South Africa or the African continent. So these leopards from the Cape, some people call them the Cape leopards, they occur in the fynbos biome, in the larger what's called Cape floristic region, because it's known for its high diversity in plants. So this fynbos biome is very similar to perhaps a Mediterranean biome. It's very dry, a lot of scrubs and bushes and a lot of succulents, and it's a lot more open than some of the tropical savannas you see in northern South Africa. What we also know is that the Cape leopard population underwent a recent population decline, a population bottleneck, we call that, and that it might be isolated from northern provinces, but this wasn't quite clear whether it was. So to put this a little bit in an overview of where these populations reside, so this is a map of Africa with all the different biomes, and you can see the Cape leopards in the very south, like I said, the Western and Eastern Cape, where the fynbos biome dominates. And we have another study site up north in the Mpumalanga province, and this is where savanna habitat tends to dominate, tropical savanna. Because of the difference in biome, you'll see that leopards prey on different items as well. Large large antelopes never made it that far south, or at least not all of them. And the prey density is also a lot lower in the south. So leopards more often hunt on rodents and hares and rock hyraxes, or very small buck, whereas up north they hunt larger prey such as impala, even up to female kudu. At the bottom, you see a very quick timeline of the evolutionary history of leopards. So the lion-leopard ancestor was about 2.5 million years ago. The origin of the modern leopard was about 2 million years ago. And you'll see at 800,000 years ago what we call the mitochondrial divergence. So we did a phylogenetic comparison of our study populations and leopards from the rest of Africa. And what you'll actually see is a very deep split of two mitochondrial clades that diverged 800,000 years ago, but these clades are represented in the northern part just the same. So we think this might relate to Pleistocene refugia when suitable habitat was contracted into small pockets. And then the out of Africa dispersal event only happened afterwards. So that was 600,000 years ago when leopards made it to Asia and started to spread across the continent. And the population bottlenecks that I spoke about that occurred in the Western Cape was in the 1600s. So this is when Europeans invaded the area and they put a bounty on a leopard's head. So there have been quite a few genomic studies on leopards done already. These are three examples on the left, but these big population genomic studies never included the Cape population, and all three of them concluded that there wasn't much population structure across the African continent, unlike Asia, where you have 8 different subspecies. In Africa, all papers concluded there's not much population structure because leopards are such versatile habitat generalists with a very high dispersal ability. There was a microsatellite study done a couple of years back, about 10 years ago, by Ropiket et al. And you can see that at the bottom right, that 3D PCA plot. And from this paper, it didn't seem like the Western or Eastern Cape really stood out. So there was never any reason to believe that this population was genetically unique, but we do know that they are phenotypically unique. So we still wondered, can phenotypic variation in this population be explained by local adaptation, or could it be because of genetic drift in the face of the population bottlenecks? So these are our first results. I'll first take you to the figure on the right. So this is the genomes that we used from previous studies, and now we added our Cape leopard population. We had 10 high sequencing depth genomes, so we aimed for 15x. And then we have 10 from the Mpumalanga. So that was that area in the northern part of South Africa. And we did a sort of continent-wide comparison. And what you see on the left, the principal component analysis, the first and the second principal components, you can really see that the Western Cape population splits out from the rest of the continent, as well as Ghana. So Ghana is what you see in that left corner. This population structure holds in the third and fourth principal component analysis, where the Western Cape population and the Ghana sit on the very right end of your figure. In the middle, we have population differentiation. So this was measured using the site frequency spectrum. So here, because we included the whole of Africa, some genomes had a lower sequencing depth. So we followed what's called the angst pipeline. So this compensates for differences in differences in sequencing depth. So we followed that pipeline to measure genetic differentiation. And what you'll see again, that Ghana really stands out. It's very high FST values of over 0.25. And the Western Cape population as well had an average FST of higher than 0.15, which is also considered significant. So here on the left, we did a population admixture plot, again with the angst pipeline, NGS angst it's called, NGS admix. So similar to the previous papers that came out of leopard population structure is that across the African continent, there's a lot of admixture going on, but the two populations that really stand out is the Western Cape and Ghana without any apparent population admixture at all. And at KS2, it's the Western Cape population that really stands out, followed by Ghana. In the middle, what I maybe also want to say, at the top left, you see the evaluation of the admixture proportions to see how well the model can explain your admixture plots. And we found that across Africa, they're not very well resolved, probably because there's a lot of gene flow happening like a lot of gene flow and litter population structure, whereas the Western Cape population, what you can see, is well resolved. In the middle, we did a neighbor joining tree based on distances. So this is a distance-based neighbor joining tree, and this is as expected, all populations sort of cluster according to their geographic location. So on the right, what you can see is a phylogenetic tree based on whole mitogenomes, and this is that mitochondrial splits that I spoke of earlier that we measured to be about 800,000 years ago. And here the Western Cape population again really stands out. In our study, we refer to it as the northern, or at least the southern and central clade. And you can see that as part of that southern clade is the Western Cape populations, but also some individuals from Namibia and Mpumalanga. So this suggests that at least since these Pleistocene refuges, there has been some dispersal in the area. So then we wanted to look at genetic load and runs of homozygosity. So these are indicative of inbreeding. So we wanted to know whether genetic drift or inbreeding could explain some of the observations we had. So like I said, we found very strong population structure and no apparent signs of recent admixture. And we wanted to know whether this could be perhaps due to genetic drift or inbreeding. So at the top left, you can see a plot where we looked at nucleotide diversity across the genome in sort of sliding windows. And you can see, although the difference is minor, you can see that genome-wide nucleotide diversity is higher in the Mpumalanga province, so up north. In the middle, we looked at runs of homozygosity, which we put into different categories. And typically, the shorter your runs of homozygosity, the longer ago inbreeding occurred, and the larger your rows, the more recent inbreeding occurred. And I think what you can see in the middle, the top middle already, is that the Western Cape population had a lot of short runs of homozygosity. And I think the figure on the top right illustrates quite nicely that the Mpumalanga province up north actually had longer runs of homozygosity. So this is indicative of more recent inbreeding. We also found a correlation between FRO and HO. So HO is observed homozygosity, and the FRO is the fraction of the total runs of homozygosity across your whole genome. So how much of your genome is covered in runs of homozygosity. So this is a negative correlation as predicted, but this was not tested significantly to be significant. And in the bottom middle, we measured deleterious load per called site. So how many deleterious mutations do you see in your protein coding genes? And what really stands out is that this is much higher in the Mpumalanga province. So this does align with our observation that inbreeding is higher in Mpumalanga, and it has been more recent because we know that recent inbreeding increases your deleterious load because these recessive mutations, they meet and become expressed. A negative effect on protein coding genes. And in the bottom right corner, we looked at the difference between whether these deleterious mutations, whether they occurred in heterozygosity or homozygosity. So I think the takeaway message from all these figures is that overall nucleotide diversity was found to be slightly lower in Western Cape, but the genetic load was lower as well, and the runs of homozygosity were much smaller. So what we actually think is that purging probably happened across generations. So because this population has been small for extended periods of time, they have managed to purge a lot of this deleterious load from the genome. So at least from this perspective, it doesn't seem that severe inbreeding is occurring in this population at all. So then we did a bit of population demographic history analysis. On the top left, we have GON. So this allows you to look at very recent changes in effective population size. And we saw that the Western Cape population occurred at about 5,000 individuals until the 1800s when there was a population crash. And this overlapped quite nicely with that bounty hunt that I spoke of earlier. So we had the Dutch settlers, then the British occupation, and after that there was a bounty hunt put on a leopard's head. And then they went through that severe bottleneck of the numbers in these programs are never that accurate, but the trends are quite clear. The recovery in the 1900s is either an artifact of the software or an actual population size, which is always difficult to tell. PCMC, this is what you see on the right. This is where you can look at effective population size more deeply in time, and you can see that both populations underwent the same demographic history, so they responded to this Pleistocene climatic shifts in the same way, which is something you would predict, seeing that they're still very close to each other. But what's interesting is that you can use these PCMC plots to measure divergence time. So this is with a software called MISTY, and that you can see on the bottom left. And what we then managed to discover from this is that both populations started diverging about 25,000 years ago. So if you keep in mind our mitochondrial lineages that we dated back to 800,000 years ago, and we now see that the Mpumalanga and Cape leopard populations diverged about 25,000 years ago. So clearly these are completely different events that happened in time and I would say are unrelated, but interesting no less. Then we wanted to know if perhaps there might be locally adapted genes that could explain the population divergence we saw. So for this, we did selection scans where you can see which genes occur at frequencies higher than you would expect. And then we did Go enrichment to see what these genes could relate to. So we found 90 genes that were enriched in the Cape leopard populations, meaning they occur in higher frequency than you would expect under neutral theory. And then we overlapped these with FST outliers because we know that regions with high FST outliers relate to selection. And then we wanted to see whether our enriched genes overlap with FST outliers. And we found that 36 of these genes did overlap with FST outliers. So this was sort of strengthen the idea that these genes could relate to local adaptation. So with your Go enrichment, you can seek out the gene functions. So a lot of them were like cranial skeletal development or cell regulation, which is quite common in these tests. But we found a couple of genes that we thought were very interesting. So I don't have time to go through all of them. So I'll just mention 5. So these were enriched genes that, like I said, they're enriched in the Western Cape populations and some overlapped with these FST outliers. So the first one is AC1, which is related to unsaturated fatty acid oxidation in mice. Then we have the GPSM1, so this is related to the accumulation of fat tissue. We have ACX3, which increases fat storage and represses fat burning during fasting. We have the SLC39, which is a critical non-compensatory role when zinc is deficient. And we have the STRA6, so this transports vitamin A in response to zinc deficiency. So these are typically what biologists found in mice during knockout tests. But what you can see from this is what we think might be the case is that because we know that the Cape Floristic region is much more nutrient poor than the tropical savanna up north, the prey densities are higher, but also the seasons are much more extremes. So the summers are much hotter and drier, and the winters are very wet and cold. So these leopards have to circulate their nutrients different than they would do up north, compensating for nutrient-poor environments. So yeah, this paper recently got published where I explain all these things in much more detail. But to summarize, the important parts to remember is that leopards in the Cape Floristic regions have adapted to a unique landscape in this fynbos biome. We have good indications that they are locally adapted. We also found divergence on these nuclear genes that we dated to about 25,000 years ago, and we have this unique mitochondrial clade that used to occur in that area and now seems to have spread slightly. So As a result of this, we do consider leopards in the Cape can be considered an evolutionary significant unit and that they should be protected as well as the environment. It's under a lot of stress in the area, and they should probably get their own IUCN Green List assessment as well to see what the population status currently is. So our results are in contrast to a lack of genome-wide differentiation found in previous studies. We found, like I said, 90 genes that were under positive selection in the Western Cape, of which 36 overlapped with FST outliers. And we found no significant differences in genetic loads and fro compared to North and South Africa. So like I said, we don't think it's genetic erosion that caused the population structure we found. But then that sort of leads us to our next leopard. Because now we want to, now we're curious to answer what could be the case with our second leopard, the red or strawberry leopard. So this is a unique color variant that occurs in northern South Africa. It has very high frequency, so 7% in some areas, and it seems to be increasing in numbers. So we were curious what drives the prevalence of this color variant in South Africa. So again, a bit of a background on where the red strawberry leopard, some people call it, where it occurs. So this is a map of all individuals that we spotted before 2022. This study is a bit older already. So these are not sightings, they're unique individuals. So you can see that the first one was in 1976. It was a hunted individual. And then the next one was spotted 20 years later, and the next one 10 years later. And currently I can see online, I can see new red leopards popping up all over the place. They even made it down to the coast. They've never been into Kruger National Park, interestingly. And what you can see is that they still mainly move in the Mpumalanga province, so that study site that I spoke of earlier, as well as in the Northwest province and parts of the Limpopo province. So in that little table with all the individuals that we cited before 2022, you can see underscored the animals that we managed to get a sample of. So for the first study, we were interested to find what causes this color variant, what makes the red leopard look the way it does. So for this, we first turned to Sanger sequencing. So we selected 6 coat color-related genes that we know relate to color morphs in different large carnivores, other cats, and we sequenced 23 exons. We also wanted to look at genetic diversity. So for this, we genotyped 20 microsatellites of 17 wild-type leopards and our 4 red leopards. We wanted to combine this with some demographic data. So we had some local mortality data, as well as some movement patterns through radio colors. So this was what we found. So we sequenced 23, and I think we finally got lucky at exon number 21. It was the last gene that we looked at, which is the TRP1 gene, and we found a stop codon that occurred in all 4 red leopards and nowhere else. And this stop codon caused a frame shift in exon 2 of the TRP1 gene. Interestingly, this deletion occurred in homozygosity with 2 other SNPs. So this is the genotype table you can see at the bottom. So like I said, the red leopard had that deletion as well as 2 SNPs that occurred in homozygosity. What's interesting is that you find 2 other genotypes that we found amongst the wild-type leopards. So at the time, we often received samples that were roadkills, so we didn't often have phenotypic descriptions of these leopards, but it would be very interesting to see what these other wild-type leopards would have looked like and whether perhaps there's some variation within red leopards as well. It could very well be that some red leopards have the deletion, but not the 2 SNPs, or like with this wild type, that you have the 2 SNPs, but not the deletion. And this would be quite interesting because this TRP1 gene relates to a cat breed as well, also in the TRP1, and then you have this is the same SNP for 2 different breeds. With the top one with the bright blue eyes, this SNP occurs in homozygosity, and at the bottom one with the brown eyes and the brown skin, this is the same SNP that occurs in heterozygosity. And we know that with the red leopards, you can see on the left, there is a bit of variation in phenotypes. Some are much more white. They have even been confused with lions, and they have these bright these bright blue eyes and this pink skin, whereas the red leopard on the bottom has brown eyes and is a bit darker as well. So it'd be interesting in the future to see what variation we see within this color variant. So we found that in our study population that 5.5% represented half siblings, and we found that the inbreeding coefficient was also relatively high. So what we think is the case. So there was some, like I said, some radio coloring data we had from biologists in the field, and it must be kept in mind that we got these leopard samples from only typically outside protected reserves. They were roadkills, they ended up in snares or were shot as a human-wildlife conflict. And this is a very disrupted system. So we found that just above 50% of leopards die an unnatural death. And as a result thereof, home ranges open up all the time. So you can see in the top left figure, there was actually one leopard who established a home range within the home range of its mother, and that's called natal phallopatry. And that's very unnatural behavior. So in saturated systems such as Kruger National Park, it is not easy for a young male to find a vacant territory and fight off the established male. This is perhaps why red leopards never made it to Kruger National Park, which is a very densely populated, saturated system. But in our study area, with such a high mortality rate, such a high population turnover, there's a lot of vacant territories, and young males don't have to disperse so far to get a new territory. This, of course, increases the chance that related individuals mate. So this could explain that such a color variant that is a recessive allele sort of takes off in a population like that, even though it is a non-fenced, it's an open system, and the leopard density is still pretty high. So we think that due to the disruptive system, genetic drift can still occur in the face of increased relatedness. All right, so then the TRP1 gene, it is typically referred to as the cinnamon locus. So it is linked to the albinistic pathway. We think that the prevalence in South Africa is caused by increased relatedness and eventually genetic drift. And then perhaps the next logical question would be, are there any deleterious mutations that are genetically linked to TRP1? So for the next study, we collected some whole genome data. So for this, we had our red leopard, we compared it to a black leopard from captivity and 3 wild-type leopards. So one was an Asian leopard, one was a African wild-type from that central mitochondrial clade, and then another one was an African wild-type from that southern African mitochondrial clade. And we know that the black leopards are actually of Asian descent, even though they occur in South African predator parks. They are Asiatic subspecies. So for this whole genome study, we particularly were interested in structural variants and high impact mutations and see how they differ between red and black leopards and the wild types. We also wanted to know whether these high impact mutations were placed in or outside runs of homozygosity, if there is a gene mutation of interest that occurs that is deleterious and occurs in homozygosity. So deleterious in this case particularly means when it's found in protein coding genes, like our TRP1 gene. It has such a dramatic effect. A lot of these deleterious mutations are known to have that. So if it occurs in a run homozygosity, so a stretch of the genome that is doesn't have any variation, then you could predict that it has something to do with inbreeding. So this is some of the results. So in the top, again, you can see that sort of comparison with the whole of Africa. This study was done before we were able to include the Cape leopard, but what you can see is that, again, Ghana really stands out, and you see that the South African population, or at least our 3 individuals, form a mixture between leopards that we found in Namibia and Zambia. But what you actually can see in the principal component analysis in the middle is that there's a very clear isolation by distance pattern, and the South African leopards that were not included in this previous study, it sort of slots right in between the other populations. And again, Ghana really stands out. If you look at structural variants only, any population structure actually disappears entirely. So this is where we looked at runs of homozygosity in more detail, so for our different individuals. So what first of all really stands out is that Asiatic leopards have much longer runs of homozygosity. We know that some of these leopards have also undergone some severe population bottlenecks in Asia, but particularly this black leopard, it's been bred in captivity for many generations to keep the black leopard phenotype sort of alive and going in captivity. So you can see how detrimental that can be. So we found that about 78% of the regions we were looking at occurred in homozygosity, which is quite extreme. If we just look at African leopards, so the red, gold, and green, we call it, you can see that the red leopards have longer runs of homozygosity actually than our wild type leopards do. So then we looked for all these high impact mutations in all our different leopards, so the red, the wild types, the Indian, and the black leopard. And what stands out perhaps is the TRP1 pops up, and in the black leopard, you can see the ASEP gene pop up. So these are the genes known to cause the color morphs. But we looked for all kinds of genes, so abnormal pigmentations, hypopigmentations, as well as hyperpigmentation. And we looked at whether they occur in homozygosity or heterozygosity. And we found another gene, myrippogene, that might actually also be related to the melanistic form of leopards, and some other genes that probably have something to do with the phenotype as well. It's hardly ever one gene that is turned on and off. There's normally different genes acting on your phenotypes that might be related to your skin type or your eyesight. So we wanted to dig a little bit deeper into what these genes could stand for. We also wanted to see whether they occur in homozygosity, heterozygosity, like I said, and whether they occur in stretches of homozygosity. And our TRP1 gene did occur in one of these stretches. So we think at least this is some evidence that this phenotype is related to, like I said, inbreeding, so the mating amongst close relatives, which just increases the chance that these recessive alleles become expressed. Like you can see here on the right, there's quite a few other ones that are of interest. That DCT seems to occur in red leopards as well, which have been found to be related with poor eyesight in albinistic individuals. So this would give a small indication that there might be some health deficits, some problematic mutations related to your genotype. That it's not just whitewashed, but that there might actually be some health deficits, like I said, with albinistic animals as well. So we also wanted to look at structural variants. You can see that the black leopards really have a lot more of these structural variance differences, clearly a sign of inbreeding. We looked at the effect that they have, so whether they are cause a frame shift or stop gains, and a lot of these genes we do consider to be deleterious. So we think that if someone were to collect data from the health state, physical state of these leopards in captivity, you'll probably end up finding quite a lot of health issues in your black leopards. And we found some other structural variants in our red leopards as well, but not more than in the wild type leopards. Yeah, so then in conclusion, we think that the size polymorphism in Cape leopards can be explained by local drivers of selection. So we think that this is in the face of nutrient scarcity, lower prey density, and more extreme seasons in this region, the fynbos biome. Whereas we think that the red leopard is genetic drift. So we think that due to increased relatedness in the area, we can see slightly longer runs of homozygosity, and we found a higher genetic load relating to genetic drift in this population. So typically when we spoke about these evolutionary significant units earlier, so typically it's advised not to translocate between populations when there's no signs of inbreeding depression and there's no need for genetic rescue, for instance, then typically it would be advised not to translocate individuals into the Western Cape. Leopards are being translocated a fair amount because of human-wildlife conflict. So conservationists always try to avoid leopards being killed when they get caught or they end up in the wrong place. So then local governmental agencies try to catch these leopards and release them in a safe place. But at least with this practice, we advise to at least keep this biome, the Western and Eastern Cape, to treat it as a separate and unique population. And as for the red leopards, I would just, this is easier said than done, but there's nothing wrong with this color morph. It's definitely a very unique leopard to have. And I think if nature is given enough space, and there doesn't seem to be, if there is no uh, strong selective drive against the color morph and it seems to take over as is, then you can just let nature be and give leopards the space that they need. I wanted to quickly thank all my collaborators. They're the ones that get me all these samples and they're the ones that have all the radio coloring data and they do all the important stuff, the leopard conservation in the field. So I wanted to just reach out and say thank you. Please try to find me on LinkedIn and any questions are welcome.
Thank you very much, Laura. I must say that I really liked your slides. I find them to be simple, effective, and also the narrative you put together. This was very didactic, so thank you very much, especially for me who, I'm not in your field, so it was great to follow you through. So thank you. Um, as I promised, I do have a few questions, and I think, well, again, my questions are coming from a place of not having the background you have, so they might be a little basic. But one question that just came actually out of curiosity, speaking of the translocation of leopards that you mentioned happens quite frequently because of human encounters. Do we know whether when animals are translocated, like how well they adapt to the not just in terms of genomics, but how well they survive, in fact, to the new place? Do we have that data?
I think there's no data. Yeah, there's plenty of people who will tell you that it has been done before and there's no issues, which I can imagine. I do think like animals are very resilient,
Mm,
particularly animals like leopards. So I think they would be just fine. But I also feel like these populations have diverged to a unique environment over so many years that it be uh, for the for the sake of the species, it would be best to keep that intact. So it has been mentioned not to translocate leopards beyond 80 kilometers. I would say just not beyond, not across different biomes. To answer your question, would this survive? I think moving a Cape leopard up north, it probably won't because of that body size issue. It won't be able to outcompete, I guess, local inhabitants. If you move a larger leopard down south, it probably will. But then the question is, does it outcompete the local leopard populations? There's currently no data that I'm aware of.
yeah, interesting observations there. Also, in terms of curiosity, so the mitochondrial clades you've mentioned, like how, because the mitochondrial genomes of mammals, it's quite small and relatively well conserved, let's say, right? Like how different are those mitochondrial clades in terms of, I guess, yeah, percentage of similarity or uh, because I like the concept of a mitochondrial clade, and I think that using mitochondrial genomes for phylogenetics and phylogenomics is very useful. And I think folks are finding that mitochondrial genomes is like certain mutations in mitochondrial genomes might actually even be adaptive, even though it's hard to prove that. It depends on the study system. But I was just wondering like how divergent those clades were.
Yeah, that's very interesting for sure. And I think mitochondrial DNA can be a bit misleading. We also did a phylogenetic study using different markers, nuclear genes and BUSCOs and UCEs, and then you'll actually see quite a different pattern for our leopard. So this is that mitonuclear discordance. These phylogenetic trees are very different. So with the leopards, if you would only do mitochondrial DNA and you see these deeply diverged clades, people would conclude they're probably different subspecies because this is a divergence you normally see for subspecies or beyond. But in fact, these can be any two leopards from Mpumalanga. These can be neighbors, if you will. So they occur in the same habitat. These are just remnants from past climatic events. So I personally don't think it represents contemporary population structure at all. It is interesting at most, and it can tell you something about the like the demographic history of a species, but I typically think it's unsuitable to use for population delineation. And the evidence that there might be an adaptive edge to mitogens, like I said, it's pretty limited. It's interesting when you look at high altitude species, it's been related to that maybe certain mitochondrial lineages survived better at high altitude. So all that stuff is interesting. I think for the leopards, I would say not relevant for adaptive variation anyway.
Yeah, it makes sense. I agree with you. Yeah, I agree that mitohaplotypes that have been linked to adaptation, it's very specific cases for very specific, biomes, like I said, altitude or even temperature as well. So I agree with you. Also, just curiosity. So, so I think there were moments in your talk in which I could understand that black leopards are all kept in captivity, but are they found in natural areas as well or not?
Oh yeah, I actually cropped it out of the talk for time reasons. Our leopard came from a predator park because we wanted to compare the Letteri's load. We wanted to look at structural variants, but the black leopard that we used is not representing the wild population at all, because there is actually a strong selective benefit uh, for the black phenotype. So you'll see that in the Cape Malay, the black phenotype is fixed now. Some areas, there's black leopards only. And these are large populations with plenty of genetic diversity. It's just that our black leopard was a good comparison because it's been bred in captivity for so long that you could really use it to look at this deleterious load and see how it compares to our red leopard. But I think typically you won't see this in the wild at all. No.
Well, I think those were the main questions that I had. And I guess just to wrap up, again, your talk was very didactic, and I think it shows, the importance of conservation biology, aligned with molecular evolution to inform our practices. I was just wondering, thinking about carnivores, as you mentioned, these animals usually, these populations have low effective population sizes, right? I know very little of them, but I was wondering if we were to compare the leopard species that you've studied with other big cats or other big carnivores, like how, I guess, safe from extinction would they be? Could we think of that comparison, like comparing to lions or other big cats, for instance?
So leopards as a whole or the population in the Western Cape?
Well, let's first talk about the population of West Cape, for instance, the one that you studied most.
Yeah, I think we were quite pleasantly surprised. We thought that they underwent this severe bottleneck, and because you don't know what you're going to get, we know that the situation of Amur leopard is very dire. There's hardly any genetic diversity left. So some of these populations have to be genetically rescued just to keep them viable because the reproductive rate has gone down. There's other forms of inbreeding depression that you see in these populations. And with the Cape leopard population, we don't see that at all. So we didn't see alarming rates of genetic diversity, which was very positive news, actually. And we think that perhaps there were more leopards left at some point than we were aware of because they were retreating in these mountainous refugia away from eyesight, I guess. So that was pretty positive. And I think the situation is looking good for Western Cape leopards from a genetic point of view. They're currently the focus by the Cape Leopard Trust is very much on establishing habitat corridors where they can, because the area is large enough, it's just very highly dense, like densely populated with humans, um, and the mortality rate is very high. So this is something that they're actively working on the ground. And then I think with a bit of awareness raising, then the Western Cape population should have plenty of space to roam. Like I said, the Amur leopard is a very different situation, and there's some other Asiatic species that are really uh, on the brink of extinction. And I think there you can see these traces in genetic diversity as well. Compared to other big cats, I think leopards are still relatively well off, even though the population numbers are decreasing, and there's some areas where they have disappeared. I think in Africa, at least, particularly South Africa, it's still the most common, one of the most, well, the most common large predator. Um, yeah,
Okay. Okay. Well, it's good then. It's good for us to end on a high note, on a
on a good note, yeah.
good note, a positive note. Okay, so I think that covers. Thanks again, Laura, for this great talk. I'm pretty sure that whoever's going to watch it from my department or elsewhere, they're going to enjoy not only the wealth of information shared, but also how you shared it. I found it to be very, very nice and eloquent. So thank you again. I will
Yeah,
stop
thank
the
you too.
I'll stop
Okay.
the recording now.